Welcome to another Explainer. Today, we're diving straight into the microscopic cellular fortress. We often think of cells as just these static, tiny building blocks, but actually, they're incredibly complex, bustling environments. And just like any well-functioning city, they need a highly secure, strictly regulated border to thrive.
Here's our roadmap for exploring this dynamic border crossing. We'll hit Section 1, the selective cellular boundary, followed by 2, passive transport and free movement. Then 3, active transport, which requires energy. 4, bulk transport via vesicle deliveries. 5 is creating the membrane potential. And finally, 6, exactly why membrane transport matters.
But before we get into the pure mechanics of it all, I want you to consider this fascinating puzzle. How does a cell actually interact with its environment without totally losing its own internal balance? I mean, it's constantly sitting in this chaotic fluid mix of nutrients, toxins, and water. So how exactly does it know what to let in, what to eat, and what to kick out?
Let's jump into Section 1, the selective cellular boundary, also known as the fortress wall.
Right off the bat, we need to understand that the cell membrane isn't just some physical rigid barrier. It has to carefully, constantly control everything that goes in and out to maintain homeostasis. Because this plasma membrane is selectively permeable, it's actively regulating a continuous flow of ions, nutrients, water, and waste. It's literally the ultimate border security, making split-second, real-time decisions to keep the internal environment completely stable.
And there are basically 2 fundamental ways things cross that border. On the left, we've got passive transport, which requires absolutely no ATP and moves substances smoothly down their gradient. Think of it like a free ride down a water slide. Active transport, on the right side, well, that's a different story. It requires ATP to push substances against their gradient, essentially charging a toll in the form of cellular energy.
Moving on to Section 2, passive transport, or free movement.
So passive transport essentially breaks down into 3 major pathways: simple diffusion, facilitated diffusion, and osmosis. And what they all have in common is they move substances down their electrochemical gradients. No direct ATP consumption is happening here whatsoever. The cell is just letting nature take its course, letting things flow naturally from high concentration to low concentration.
Let's start with simple diffusion. This is the path of least resistance. It allows small, lipid-soluble molecules to slip right through the barrier without needing any transport proteins at all. Molecules like oxygen, carbon dioxide, and steroid hormones, they don't need a special gate. They just naturally move from an area where they're highly concentrated to an area where they're less concentrated, slipping right through that hydrophobic lipid bilayer.
But what happens when molecules are too big, or they just can't cross that lipid bilayer easily? Well, that's where facilitated diffusion comes in. They use specific channels or carrier proteins like GLUT for glucose or aquaporins for water. Now, even though the cell is using these specialized proteins to facilitate the movement, it still doesn't cost the cell any direct ATP. Why? Because the cargo is still moving right along with the flow, from a high to a low concentration.
Now, let's look at a very specific and super important type of passive transport, osmosis. Basically, water moves naturally to dilute higher solute concentrations, and depending on the tonicity, this can actually cause a cell to swell or shrink. If the outside environment is hypotonic, meaning it has fewer solutes, water rushes into the cell. If it's hypertonic, water rushes out. In an isotonic state, things are equal, so there's no net movement. This natural balancing act is entirely passive, but absolutely crucial for the cell's survival.
Let's shift gears to Section 3, active transport, which requires energy.
Take a look at these numbers. 3 sodium ions pumped out and 2 potassium ions pumped in. This specific ratio is the absolute hallmark of the classic sodium-potassium pump. It burns 1 ATP molecule to enforce this exact exchange. This right here is primary active transport in action. By forcing these ions against their gradients, literally pushing them from low to high concentration, the cell maintains vital things like its volume and its ability to fire neuronal signals.
And beyond just sodium and potassium, ATP-dependent pumps also heavily regulate calcium. And regulating calcium is completely non-negotiable for things like muscle contraction and neurotransmitter release. The cell actually has to maintain very low cytoplasmic calcium levels, so it aggressively pumps calcium out of the cytoplasm or hides it away in the endoplasmic reticulum, burning up ATP to keep that precise, delicate balance.
But here's where it gets really interesting. Secondary active transport is kind of like cellular carpooling.
It uses the energy that's already stored in a primary ion gradient to pull other substances, like glucose, along with it. So instead of burning a fresh ATP molecule right at the transporter, it uses the momentum of something like a sodium gradient. Whether it's symport, meaning they're moving together, or antiport, moving in opposite directions, it is a brilliant energy-saving mechanism for moving tough cargo against the gradient.
All right, Section 4, Bulk Transport, Vesicle Deliveries. Sometimes the cargo is just way too big for a pump or a channel. That's where endocytosis comes in, and it's fascinating because the cell physically wraps its membrane around large materials, literally eating bacteria or drinking fluids. We call the eating part phagocytosis, which handles large particles like cellular debris. Then there's pinocytosis, which takes in extracellular fluid and dissolved molecules. And finally, receptor-mediated endocytosis is highly specific. It allows the cell to efficiently snatch up very specific targets, like LDL cholesterol, right out of the environment.
Now, let's look at the exact opposite process, exocytosis. When it's time to export large materials, like hormones or antibodies, exocytosis follows this highly organized assembly line pathway directly out of the cell. Step 1, the cargo is packaged in the Golgi apparatus. Step 2, it's shipped in a secretory vesicle. Step 3, it fuses with the plasma membrane. And Step 4, it's finally released into the extracellular space. It's shipping and receiving at a microscopic level.
Which brings us to Section 5, creating the membrane potential, the electric grid. So, because ions carry electrical charges, throwing them unevenly across this cellular border creates a literal battery inside your body. We call this the membrane potential. By constantly regulating ions like sodium, potassium, calcium, and chloride, the transport mechanisms we just talked about are actually creating a distinct electrical charge difference between the inside and the outside of the cell.
The crucial question here is, why do we even need this battery? Well, without this microscopic electrical grid, your brain couldn't send signals, your muscles couldn't contract, and your senses, they'd go completely dark. This membrane potential is absolutely essential for every single neuronal signal, muscle contraction, and sensory response you experience.
Let's wrap things up with Section 6, why membrane transport matters in the real world.
Understanding these pathways isn't just textbook biology, it is completely revolutionizing biotechnology and medicine. It allows scientists to literally hack the cell's own transport system for things like targeted drug delivery and gene therapies. By studying how receptors and ion channels work, pharmacologists can specifically target them to fight disease. We're at a point where we are designing nanoparticles to trick the cell's transport mechanisms into pulling life-saving drugs directly inside. How wild is that?
I think this quote perfectly summarizes our explainer today. The cell membrane is not simply a barrier. It is an incredibly dynamic, highly selective transportation system that controls what enters, what leaves, and how cells maintain their internal balance. It keeps you alive at the most fundamental level. It's not just a static wall. It's a bustling port of entry, complete with its own microscopic customs agents, toll booths, and massive cargo ships.
So, I'll leave you with this thought to ponder, because the future of medicine is quite literally waiting right at the gates of the cell membrane. If we can master and engineer these microscopic cellular gates, what currently incurable diseases could we conquer next? The possibilities really are limitless. Thanks so much for exploring this complex cellular world with me today, and I'll catch you in the next one.